Hello, everybody. My name is Dr. Ali Yahya. I'm the Director for Marketing at Flexion Biosciences. Today, I would like to introduce Dr. Christian Ionescu Zanetti, CTO at Flexion. Dr. Ionescu Zanetti is the Chief Technology Officer and co-founder of Flexion Biosciences. In this role, he is bringing discovery platforms from concept to product. Flexion's 3 technology platforms are used in a range of preclinical drug discovery, clinical research, and diagnostic applications. Christian received his PhD in biophysics from UC Santa Cruz. Today, his talk is implementing ultrasensitive liquid biopsy workflows. Now, for questions, there is a box on the side of the software. If you have any questions, please write it down in that box, and Christian will read it at the end of the talk. Christian, it's all to you now.
All right. Hey, everybody. Thanks for joining us. Just a few things before we start. The talk will be about 45 minutes. We'll leave some space for questions at the end. And as Ali told you, you'll be able to type those in, and we'll address as many of them as we can. Now, on to the talk. We'll be telling you about our unique approach to ultrasensitive liquid biopsy variant detection using ERASE-seq, the variant caller that we've developed, and this molecular amplification pool approach. So the overview of what we'll be talking about is as follows. So first, we're going to talk about the great promise of liquid biopsies briefly. We know what that is, but then focus in on the challenges to making those a reality. So the current approaches and some of the commercial tests that are out there and where they fall short. Then we'll talk about our approach, the molecular amplification, using molecular amplification pools and using a novel statistical based caller that achieves great performance without the use of UMIs. And then a few case studies for the implementation of our technology. Finally, we'd like to make this very practical and tell you a little bit about what we found to be helpful in terms of actually implementing these types of assays in your own lab, what we found to be the critical quality metrics, and just how to go from A to Z with a new assay. So first of all, liquid biopsies, as you know, are a hot topic. They've been showing a lot of promise, and they hold there's great hope that they can not only supplement but eventually replace solid biopsies. Solid tumor biopsies have been around for a while. They've been used to guide treatment and diagnose cancer. Of course, the issues with them are that you can only typically biopsy a patient once, and the cancer is a changing disease that adapts to treatment. So it's very hard to figure out what the changes are in tumor biology over time and tailor treatments accordingly. The other big challenge is, of course, diagnosing cancer early enough. So as you probably know, a lot of cancers are diagnosed in late stages where the options for treatment are limited, and it's very hard to make a difference in terms of the longevity of the patient. And so detecting cancers earlier is a huge area of study, and if we can pull it off, it would be great for improving human health. Now, just a couple of definitions before I move away from the slide. When we talk about liquid biopsies, we're talking about generally either cell-based biopsies, meaning circulating tumor cells, or circulating nucleic acids like DNA and RNA. There are newer analytes like vesicles that are being analyzed, but most of the talk here I'll be focusing on circulating tumor DNA, and people refer to it as CF DNA, meaning cell-free DNA. That's present in the plasma of the patient. It's comprised of ctDNA, which is the circulating tumor DNA. So it's actually strands that are originating from the solid tumor, plus a lot of what's normal tissue DNA. So DNA that's originates from normal cells that are not part of the tumor that do die and shed nucleic acid into the bloodstream. So that's an important distinction, and I'll try to be clear when I use those two terms, but it often causes confusion in the literature, and I just wanted to put that up front. I think, as you'll see, the ratio between these two analytes, the ctDNA and the normal tissue DNA, is what's important in terms of being able to detect tumor-derived variants in the blood. So now, after the promise, I'll tell you a little bit about the challenges of doing this. We found that a lot of cancers do shed DNA into the bloodstream, which is great. This study found over 75% of samples across a number of different cancers at different stages were positive for ctDNA, circulating tumor DNA. However, there are significant differences between both the different indications and the different severity of the cancer. So what people have found, and this is just a publication from Science Translational Medicine, there are a few publications on this now, is that as you increase, as the severity of the cancer increases, meaning larger tumors and metastatic cancers, you have more and more DNA in the bloodstream. That makes a lot of sense. So a lot of the studies you see out there have a mix of patients that's typically biased towards stage 3 and 4 cancers, where these variants are easier to detect. But even in those populations, as you'll see, the variant allele frequency, so the ratio of, which is driven by the ratio of ctDNA to cfDNA, is quite low, often below 1%, and that's what presents a challenge in terms of sensitivity of the technique used. Now, there's another challenge besides the percent of tumor DNA that's very important to understand, and one is the low amount of DNA input available. So when the labs we've worked with, people typically get between 1 to 30 nanograms of DNA from a blood tube. Typically, and this depends on a number of different variables, but this really dictates what the lower detection limit is. So if you have a low percentage of ctDNA present, and people often see percentages for the allele frequency of the mutations all the way down to 0.1%, you need a large number of starting copies to be able to detect that. So, for example, if you're in a good place and you've got 30 nanograms of DNA, you have just 10 mutant copies supporting a 0.1% allele frequency call. So you need very high efficiency, and we'll see why this is important in a second. You need to be sure that all of your molecules enter the sequencing reaction. If you're at just a third of that, 10 nanograms, that's pretty much your limit of detection for 0.1%, because at 10 nanograms you only have 3 mutant copies supporting that call. And if you have lower quantities than that, you'll still be able to run your experiments, you still may be able to detect variants, but you'll be limited to those variants that are above, say, 1%. So the next question you might want to ask is, well, what are these allele frequency variants for tumor-derived somatic variants that you see in blood? And we've got now extensive experience through working with a few clinical labs out there where we see a variety of somatic variants, and then we can as you'll see in a second, verify that those variants are truly there using orthogonal techniques. But the bottom line is that a lot of those variants are below 1%. A majority of them are below 1%. And in fact, there's about 30% in this population that we've looked at in lung cancer that are between 0.1 and 0.5%. So it becomes critical for the success of the assay to be able to have to detect variants in this, let's say, 0.1 to 1% range, and with high accuracy and low false positive rates. So now I'm going to talk a little bit about what the current prevailing approaches are to address this problem. And basically, most of the commercial tests that are out there, and a lot of the tests that are being used in research studies, are using unique molecular identifiers, also called molecular barcodes, which are basically it's a very elegant technique that allows you to essentially track each starting molecule from the cfDNA sample all the way to the final reads. And so the idea and eliminate a lot of the errors that happen in the process. And this addresses both amplification errors as well as sequencing errors. So you've probably seen this kind of pictures before. This is adapted from one of the Illumina brochures, but basically you have this molecular barcode sequence. It gets attached to your input molecule. Now, what's interesting here is this happens to be an amplicon derived molecule. So actually, this is not the starting cfDNA molecule. It's the first replication of that. And then this goes through all the subsequent PCR-based amplification. It creates a number of molecules that are all in the same tube. And then so in the process, errors get introduced. Then this is sequenced. And all of these, using the tag, using the unique molecular identifier, all of these are collapsed into what's called a consensus sequence. So in this case, you have some, say, 10 reads, and then you end up with one read where you achieve consensus by saying, okay, well, we know this is all coming from the same starting molecule, therefore all of our reads should be identical. If there are any differences, those must be errors, and then you use either quality metrics or statistics to determine which is which. So you come up with a consensus read, and this has been shown in analytical studies to do a great job of error reduction, and as a result has been adopted by cell-free DNA companies. Now, what we see in practice is quite a bit different than what we see in this early analytical work. And there have been now a few commercial tests that are out there, and they've presented studies showing very promising performance in cancer, but there have also been a number of independent studies looking at the orthogonal performance of those techniques. So basically taking sequencing-based studies, sequencing-based approaches that are lab-developed tests, and comparing the results to one another from the same exact starting sample. And this is a very good study that was done by AstraZeneca, where they took 4 such tests. They had 24 patient samples, and they basically sequenced the healthy tissue. They sequenced the tumor from these patients, as well as all of these liquid biopsy results. So as a result, they're able to determine with high confidence which variants are true positives, which variant, and they're depicted here in blue, which variants are false positives in red, and the false negatives, so the missed variants that influence sensitivity are in orange. And if you look at this, now I should point out that this is a challenging population, so it's not necessarily very late stage cases, but they're definitely, it's a representative diagnosed population, not pre-diagnosis. And if you look at this, you see that we still have quite a ways to go when using this current state-of-the-art technology with well-developed commercial tests before we can confidently detect variants below 1%. So above 1%, you have a number of tests that perform really well. They don't have false positives. They don't have many false negatives. Below 1%, that changes, and you basically see, well, a little bit of everything. You see, false negatives, false positives, and so all I want you to take away from this is that we still have quite a ways to go, and that the idea behind unique molecular identifiers doesn't quite work as well as we thought in practice, and we have some ideas about why that is. So just putting numbers around those figures that you just saw, you've got sensitivities anywhere from 40% to 90%, but then the positive predicted values, which are very important for using those variants that are detected in the clinical decision-making process, are again anywhere between 50% and 80%. So we can improve on this, and I think what's important to note is that even with a lot of development time and money, it's hard to make these assays work perfectly. And we also tried some of these assays to start in our lab that were based on barcodes, and we found that there are reasons why those shortcomings come up, especially in cell-free samples. So what causes this difference between the ideal performance that's very low and the real-world performance that you just saw? There are a few factors that are important. So one is each one of these steps, which work perfectly on my slide, can have errors and issues that crop up in a real-world setting. One is the attachment efficiency of the molecular barcode itself. So often this is quite low, and this is why things like dual barcoding don't work at all in liquid biopsies. And if you have something like a 20% attachment efficiency, you won't have a problem in a contrived sample where you start out with a lot of copies. So let's say you have, a few hundred mutated copies, then in a high background you won't have a problem. But if you have a low number of copies and you have only 20% attachment, it's really easy to lose sensitivity by just never having those mutated copies never enter the reaction. The other issue is that you're often not really tagging the initial starting molecule. So a lot of those approaches, at least if they're amplicon based, rely on a first amplification of the molecule before the tag is applied. So this, if this is not truly the starting molecule, then you still have some small, some chance that in the first round you introduce errors. Another issue that's important is that during the amplification, errors get introduced in the sequence itself, but other errors also get introduced in the tag, because the tag, of course, is getting amplified as well. If you have random tags, it's relatively easy for an error in the tag to cause basically jumping of molecules from one family to the other. So that's another way that errors can be introduced. Finally, some of the workflow issues that people run into. One is the need for super high read depth. So if you, again, thinking about the numbers, if you are trying to detect variants at 0.1%, you have to have 1 in 1,000 mutated reads. And if you want, that means you need a minimum of 1,000 read depth in this at the consensus stage. That means that if you want 10 reads supporting each consensus read, then you need a minimum bare minimum depth of 30,000x just to have 3 reads supporting the call. And that, just inflates the requirement for high read depth and inflates the cost of these assays. Finally, for individual labs trying to implement this, I've seen more than one where at the UMI bioinformatics stage was the actual bottleneck for this. So there's a complexity and a time and effort required to properly implement these methods, and that's often where things become difficult. So given all that, we started looking at doing things a different way. And what I'll be talking about for the rest of the talk is our approach called erase-seek, and that just stands for elimination of recurrent artifacts and stochastic errors. So what we did was we started looking at, without the use of UMI, the targeted we used a targeted sequencing panel that's about 30 kb long, and we just started looking at error profiles. So we started using a very sensitive caller without really having any quality cutoffs, and just looking at just letting everything through and looking at all the false positive calls that you could get. And what you see is if you plot it versus allele frequency, you kind of see what you expect. There's a lot of noise below 0.1%, but then a fair number of calls between 0.1 and 1%. And then the challenge is figuring out how you can extract most of the information out of this data sets. And what we started doing then is working really hard on running a number of normal samples and figuring out where those errors are coming from. And what we were able to do by comparing all these different runs is really divide the errors into 2 different overarching classes that are fundamentally different. One class is stochastic errors, meaning errors that are randomly introduced, let's say polymerase errors that are randomly introduced throughout the sequence, the coverage area, and those will not be, the characteristic of those is they're not repeatable sample to sample. So if you have a random error happen, early in the amplification cascade, you'll see something that looks like a real variant. You also have what we would call recurring artifacts, and those can be due to a number of reasons. One is start and stop of amplicons. There are homopolymer regions. There are even bioinformatic, common bioinformatic issues that crop up, and those are going to be to some extent common between different runs. So as you can see, looking at this graph, errors above 0.3% we found to be overwhelmingly recurring. So repeatable if you run enough normal controls. Errors below that we found to be stochastic. And what we started to do with all of this data is to assemble what you would call a background model. So in other words, for each position or variant on the panel, and there are about 90,000 such positions, you just saw them here, we would look at all of the reads, all of the variant reads, and assemble basically a noise profile across the full panel. So what you see depicted here, and this is just an artist's rendition, is a mean allele frequency for each position and a variance around that mean frequency. So that's the first step to implementing our caller 2, as you'll see. And then to that background model, we would compare the sample data. So what you see here in red is an example, again, artist rendition of what a specific set of sample calls might look like. And they may or may not be within the error distribution. So we then devised a statistical test or a set of statistical tests to figure out what is the probability that this data point is really part of the error distribution? And we used the binomial test for that. And what we came up with, the result of that is a confidence score, essentially a probability that that specific variant is part of the noise distribution. So what you see in a real world case, and then this is the same experiment that you saw earlier, is that if you, instead of plotting allele frequency versus variant position on the x-axis, you plot this, what we call confidence score, but basically the probability that the variant is not part of the noise distribution. You see much better differentiation for a small number of variants, and essentially this is what we use to call true positives, as opposed to just allele frequency, which is what most variant callers do. So then, if you plot this back onto the allele frequency versus position graph, you see something like this, and this happens to be a analytical sample, but you see how these known variants are now identified using this confidence score, whereas below, by just drawing a minimum allele frequency line through this graph, you would get a lot of false positives and a lot of false negatives, and you really wouldn't be able to go down to 0.1%. You might be fine at 0.5%. So this is essentially how we apply our caller for existing data sets. Again, this point here is just one sample data set, and then for panels that we know, we already have this error profile figured out. So doing that wasn't quite enough. So it improved our caller a lot. We could be able to detect things in the sort of 0.2 to 0.5% range, but it was hard to do that across the full panel. And we could do quite well across a limited number of variants, but we wanted to improve that further. So the next step that we took was to come up with this concept of molecular amplification pools. Essentially, the idea here is that instead of tagging each molecule at the onset, what you do is you would divide the starting sample into two different sets, two or more. And then you would, and this is just physical division, these are two different tubes at this point, and then you amplify each tube separately. And the power of this approach is that any random errors that appear in one molecular pool are very unlikely to also appear in the second molecular pool. So it allows us, whereas the background modeling allows us to remove recurrent artifacts, this approach allows us to remove stochastic errors that are random. And the reason for that is there are 90,000 positions. A truly random variant that originates early in the amplification process in one pool is very unlikely to also happen in the other pool, and in a situation where each pool has only a few thousand molecules. So what happens is we amplify all this, we tag it, we tag each pool, and that's basically using index tags. So at this point, these two pools are just being treated as like technical replicates, and then they get sequenced together. And basically what it does in my artist rendition is this adds a second data point to this graph. And so now with our statistical confidence score, we can ask the question, you've got distribution A, which is the blue, and then you've got distribution B, which is the red. How likely is it that both of these are part of the same distribution? And that gives us a lot more confidence. And for example, here you would say, okay, now I'm sure that this is part of the noise, but and I'm sure that this is also stochastic error, but these ones are further differentiated from the original error distribution. And if you plot this in terms of error rates as a function of allele frequency, what you see is that you do much better with respect to standard callers, even for even using one molecular amplification pool, basically reanalyzing the data using a background-aware caller, but that really the best gains are we use at least 2 amplification pools, and in the 0.1% range, you really need to do that to get high accuracy. So I want to show you now some of the analytical results and some of the clinical results that we've obtained using this technique. So analytical testing, I'm just showing you this because we just spiked tumor DNA into normal DNA and then did the sequencing. We have got variants all the way down to the 0.1 to 0.5% range, a lot of them in this range, and we obtain very nice sensitivity specificity, meaning in this critical range of 0.1 to 0.5%, we have about 95% sensitivity, and then we get only 0.1 false positives per 10,000 variant tests for the best implementation of this erase-seq caller. Now, we've used it with our own panel, but we also can adapt this to other panels like the TrueSight Tumor 15 from Illumina, and you have similar gains. The performance won't be the same, but the gains with respect to unreferenced callers are the same. And then this is a comparison to some of the other approaches out there. There are quite a few approaches that show detection, low limit of detection in the 0.1 to 1% range. However, oftentimes the specificity is quite a bit worse. And the way that people deal with that is by severely reducing the caller space. So oftentimes, to achieve the high sensitivity, you have to only look at, let's say, about 200 different sites across the panel. And I think that's a good approach, but it negates some of the advantages of sequencing for being able to go far and wide across and detect many different variants. So what's most important though is the clinical testing. So what we've done here is we've used our technology on sequencing data and compared it to orthogonal ddPCR testing in a lung cancer population in collaboration with a clinical center. And what you see here is that compared to ddPCR, we get very, very good both sensitivity and specificity. And a lot of the, as I showed you, a lot of the variants that we see are in this below 1% range. So I think this is great, and I think more of this needs to be done. In other words, orthogonal testing is critically important because it sort of tells you that your sequencing technology is seeing what's really in the blood, and it's sort of making you immune to biological issues like tumor heterogeneity or shedding of DNA into the blood. So with that, I wanted to go into a couple of case studies that we've done that are really show how this technology can work in the real world. So case study number 1 actually is where we got this data set. It was a cancer center that wanted to improve their sequencing accuracy, and they were doing this in lung cancer patients, and we're using ddPCR as well to look at the few key variants that were clinically actionable. And we used exactly what one should, which is, in our view, is the MAPS approach and erase-seq. So we had multiple data points for each sample, and we got very good sensitivity specificity with respect to these droplet digital PCR assays. So again, these assays have very high sensitivity down to at least 0.1%. We had T790M that was they were using Bio-Rad ddPCR as well as beaming. We have L858R, which is a sensitizing mutation, and there again ddPCR and Bio-Rad was used, and the LX19 had I think 3 to 5 different deletions that were covered by droplet digital PCR, and we compared all those directly in matched samples, and this is what we got with respect to erase-seq used on a targeted 31 kb panel that's pan-cancer. Now, what's important is where we do see the discordance. For example, we have a PPV of 91% here with respect to ddPCR, so we had some discordant calls over the full range, which was 348 diagnosed lung cancer cases. Now, of these 7 discordant calls that we found, 4 were actually supported by tissue. So I think our conclusion from this was at these locations we had similar sensitivity specificity to PCR with respect to what's really in the blood. Now, the power of sequencing, of course, is to be able to detect additional variants that are clinically important but aren't covered in narrow PCR panels. So what you see here is focusing in on a particular class of variants that are known to be important for TKI, either sensitivity or resistance. So this type of therapy is administered where patients either have a DELX19 variant or L858R. Those are called sensitizing variants, and they can be found for this cancer center would administer it whether these were found in either tissue or blood via this assay. On the flip side, on the resistance side, the T790M variant confers resistance to these drugs. And so we wanted to detect that. That's important because it would cause you to perhaps switch the patient to a different therapy or choose a different type of therapy to begin with. What's important to note here is that there are a number of other variants across the EGFR gene that are important to this drug class, and using our technology, we were able to detect all of these types of variants, not just the very common ones, the 3 very common ones that are covered by ddPCR. So in a significant portion of patients, we were able to improve the detection of either sensitizing or resistance mutations by using sequencing. And we know these variants are accurate because we have the orthogonal determination from ddPCR. So this is very exciting work that's being reviewed now as a publication, and I won't go too deep into it, but you can also draw a lot of conclusions about the cancer progression, because there are 2 different cohorts. There's a cohort that's being tested at diagnosis, and there's a cohort that's being tested at progression. And anyway, it's a very nice data set. So I'll move on to the second case study, which is applying this array-seq technology to existing data from an NGS panel. This is interesting because it's a very low barrier to adoption of array-seq. So basically, this is a lab that was using a Acyl-Amplicon panel from Swift Biosciences, which is very nice because it has full coverage of the TP53 gene, unlike other panels, and they're looking at trying to detect as many TP53 variants in ovarian cancer as they could across the TP53 gene. And so they had this data, and they said, okay, well, can we apply array-seq on data that we've already analyzed using standard callers to improve the calling accuracy? So in this case, I'm really only using half the power of array-seq, but it still can provide great gains. We're just using the background correction piece. So here's the results. So what they had originally done, and I hope you can all see this, is just use a low-freq caller, which is a good standard caller for looking at low allele frequency variants from deep sequencing data. So they had data that was at high depth, over 5,000x per sample, and they had sequenced both the cfDNA, the tumor, and the tissue. So the tissue provides a nice sanity check where you can really determine which tumor variants are somatic and which are not. So from the same data sets, we reanalyzed, all of the data for the sample set. So again, we reanalyzed the tissue, the tumor, which you expect to see the same things, and we do. as the standard caller, and then the cell-free DNA. Now, what you see is in cell-free DNA, we were able to detect significantly more, basically variants that are present in this, again, 0.1 to 1% range. So in this 5-sample data set, we detected 1, 2, 3, 4 variants that were previously undetected using standard callers. And then the folks we were working with went out and tested these via droplet digital PCR, again, as a confirmatory test by designing specific probes. And we're able to confirm that these are indeed present in the blood. And they, for a good number of them, they line up to tumor somatic variants. So this is an example where, from existing data, people were able to use our technology and get better results. And it was a really exciting study. And then I'd like to finally talk about a third case that's still ongoing. That's an exploratory study aimed at detecting recurrence signatures in melanoma. So the challenge here was that they wanted to adopt our technology, and that necessitated the new panel design. And so here we were working with a collaborator called CellWorks Group. And what we do here, and what we would do for similar projects, is to design, de novo design a new panel. In this case, we designed a 700 amplicon panel covering 49 genes. Our typical pan-cancer panel is about half of that, about 300 amplicons across 50 genes. So the way that this panel design is done is to maximize patient coverage. So what we wanted to do is, as you include more and more genes and more amplicons, you get better and better patient coverage. If you know where the variants are that are typically found in the population. And so in this case, our target was to achieve 90% patient coverage. And this is an ongoing project where we've started patient recruitment. So finally, I think what I want you to walk away with is that despite the prevalence and the good analytical success of UMI solutions, there are significant shortcomings when trying to apply those solutions to cell-free DNA and other liquid biopsy samples where we have limited sample input and very low allele frequencies. And even in commercial testing, for tests that are really well developed, there's still a ways to go to achieve good concordance between those tests, sub 1%. As an alternative to that, we propose using molecular amplification pools, where you obviate a lot of those technical issues that come along with UMIs, and you have an ability to then eliminate stochastic errors and eliminate recurrent errors by using background models. And we have about 95% sensitivity down to 0.1%, and we don't just show that in analytical samples, we show that in real world cancer patient-derived samples. And orthogonal testing basically shows performance that's on par with our analytical testing, and has been used to detect a large number of actionable variants in patient samples. Now, I think one of the things that sets our approach apart is the low barrier of entry. So I'm almost done here, but I wanted to spend just one slide talking about the implementation of this erase-seq technology. So we have a few panels that we've already validated, meaning we've already built the background models that I've talked about. And for those, we actually have a client that can be used to run the bioinformatics. It just takes in your FASTQ files and then returns back VCFs. That's very easy to use, easy to implement, and it doesn't require you're building up a pipeline in your end. Now, the other cases I talked about were cases where people want to apply this to a new panel that's an existing, new to us, but that's being used. What we need there is the replicate runs of normal or DNA standard samples at a minimum of 500x depth to build a background model, and we will do that and add it to the application, and then you'd be ready to run your data sets. So right now we've just launched this client, so we're offering people a free trial of the technology, either the use of the caller or even the development of a new background model. So it's a great time to try out this approach if you're working in the liquid biopsy area and have data sets that apply. So with that, I'm going to end the talk. If you want to pose any questions, please feel free to do so in the chat screen or the question screen. And if you'd like to set up a trial or need any other information, you can contact us as well. So thanks for your attention. So let's see. There are a number of questions. One question is, how long does it take to develop the background model? If we have all the data, it takes us about a couple of weeks to develop and validate the background model. There's another question regarding duplicates. So regarding chemistry, what is done differently with the maps rather than running duplicates of your cfDNA sample? So the answer to that is we've introduced maps as a concept for error elimination. Essentially, what we're doing is running technical replicates. So if you run technical replicates, that's similar to running maps. And that data can be fed into the RACI caller and you know, used with the same kind of statistical approach. And then there's a question about PCR duplication. So saying, does PCR duplication affect the allele frequency calculation here? I think the answer to that is probably, but we don't take into account PCR duplication. So if you're individually tagging molecules, you may have some understanding of the amount of PCR duplication. In this case, because we're using a panel, you know, all of the starting molecules go into the same pool. So whether our variant comes from, you know, 1 or 2 starting molecules, we don't know, but we can tell that it's with high confidence whether it's a significant presence or not of that particular variant. Let's see. Okay, I've got quite a few different questions coming up, and I'll try to get to them. There's a question about slide deck availability. I can make that available. I don't think we'll have it online. I believe we'll have the webinar recording available to people that registered. If you'd like the deck, I can send it to you privately. Just send me an email. I'll send it to you, the person that asked. But if there's somebody else that would like it, I will just contact us. What's the sequencing depth required to detect 0.1% variance, and how does that compare with duplex UMIs? That's a good question. So right now, our sequencing requirements in terms of depth are 5,000x per molecular amplification pool or per technical replicate. So the total sequencing depth requirement to detect 0.1 is 10,000x per sample. I think that's significantly less than the depth required for duplex UMIs, which I believe is over 30,000x, but I'm not sure. But I'm pretty sure about it, but it's not 100% sure. Let's see. There's another question regarding target capture data, and I think that's, and the question is, can you create background models for targeted capture data as well, or only for amplicons? The answer is yes. We haven't done a lot of that work. So amplicons are a little bit easier because every molecule, every read covers the same the same region in the genome. So you have a guaranteed uniform depth at each position on your panel. However, within the same amplicon rather. However, if you use a targeted approach, you run into, at the endpoints, you might have different coverage, away from the probe, but I don't believe that's an unsurmountable issue. So I think the answer is yes, that we can create background models for target capture data. Let's see. I think that's about all we can get to now, unless there are other questions. One other thing I wanted to point out before we conclude is that there was another question about the maps and statistics and replicates. So I think there are quite a few, the idea of running technical replicates, certainly not a new one in science. So I think people do run sequencing replicates. The difference with the maps concept is that we have created a statistical background based on, based on binomial distributions that, while it has been used before in things like expression profiling and deciding whether there's a difference in expression or copy number between different genes, it hasn't been really applied to mutation detection. So the difference, so this is significantly superior to just saying, okay, I'm going to run, replicates and then, use a little frequency threshold in both replicates and then decide, okay, well, do a union of both calls. I can tell you, because we've done direct comparisons with that type of approach, and by using the correct statistics and building up the background model to feed into those statistics, you get much better performance with this MAPS approach. So it's not, so the analysis really integrates both all of the background information and all the sample information into your results. All right, with that, I think we're going to adjourn. Thanks a lot for your time. I appreciate it. I'm glad to see that we're getting done before the hour. And if you think up any new questions, or you want to, something comes up after the talk, feel free to contact us. And yeah, have a good one. Happy Thursday.